they are the technologies which are used to help the couples to reproduce. So that is why it is called as assisted reproductive technologies. The fertilization and the development of the embryo or the formation of the embryo takes place outside the body of the mother. The babies are also called as test tube babies. The zygote will not be of 8 cell stage, but it will be of either 16 cell stage or 18. So when it is that condition, it will be directly put into the uterus. So that is called as IUT. Hello everyone, a warm welcome to this session of concept class for class 12th biology. I'm Dr. Divya, biology faculty. So in this session, which is the last session of the chapter reproductive health, we will learn about assisted reproductive technologies, wherein if the male and the female partners are infertile, that is, they are not capable of bearing a child, what are the different technologies that assist in reproduction or fertilization process is what we will see under this particular session. So assisted reproductive technologies are very important because it helps the couples who are infertile or who are not in a position to bear a child. They help them to bear a child. So therefore, the couples could be assisted to have children through special techniques commonly known as reproductive technologies. Assist means they help. So that is why, what do they help in? They help in reproduction. So that is why they are the technologies which are used to help the couples to reproduce. So that is why it is called as assisted reproductive technologies. Now in this assisted reproductive technologies, there are various methods that are followed. One is in vitro fertilization, that is IVF, then intrauterine technologies, that is IUT, then intracytoplasmic insemination, that is ICIS, then zygote intrafertilization technology, then gamete intrafertilization technology, and artificial insemination, that is AI. So if all these technologies fail, or if the couples do not have enough money to undergo these technologies, then there is one more option, that is adoption. So adoption is nothing but wherein the couples, that is through government agencies, they can adopt a child through legal procedures and all that, once all the formalities which or all the clauses that are put forth by the government is met. So that is nothing but legal adoption. So talking about each of these technologies one by one. So first we'll talk about IVF, which is in vitro fertilization. In vitro. So there are two terms. One is in vitro and one more is in vivo. So in vitro and in vivo. In vitro is in the laboratory conditions that it is done. In vivo means it is performed within the organism itself. So that is called as in vivo. So in vitro means what? It is done in the laboratory. So what is done in the laboratory? The name itself says in vitro fertilization. So or in other words, if I have to put, it is nothing but the fertilization that takes place outside the body of the organism and followed by embryo transfer. So once the fertilization is done, that is the egg from the female is retrieved and the sperm from the male is retrieved, and in the laboratory they fuse between the egg and the sperm and they keep it under suitable conditions or controlled conditions in the laboratories, and once they develop into the zygote, that zygote is inserted into the uterus of the female, and we know that the zygote will go there and embed in the endometrium wall of the uterus of the female, and the development of the fetus will start there. So that is in vitro fertilization. So this method, because the fertilization and the development of the embryo or the formation of the embryo takes place outside the body of the mother, the babies are also called as test tube baby. So this method is commonly or popularly it is known as test tube baby program, in which the ova from the wife who will be the donor. So donor is the one who will give, receiver is the one who will receive or will take it. So wife who will be the donor, and from the husband who will be the donor again. From the wife what is taken? Ova or egg is taken. From the husband what is taken? The sperms are taken and they are collected and they are induced, that is they are fused or fertilized to form the zygote under simulated or under controlled sterilized conditions under the laboratory. So what they do is here, they collect the sperms from the donor male and they collect the ova or the egg from the donor female. They are brought into the laboratory. In the laboratory, they take a petri plate or a petri dish, and in that suitable nutrient media, whatever is required will be there, and under simulated conditions, they fuse the sperm with that of the egg and zygote. What will it develop into? It will develop into the zygote. That zygote or the next stage, that is the morula stage that we call, no, or the blastomere, 16 or 18 celled blastomere, that will be inserted into the womb. That is, this is the uterus. It will be inserted into the uterus or the womb of the mother, and there it will go and embed where? In the endometrium lining of the uterus, and further it will start developing into the fetus. So this is called as in vitro fertilization. Why is it called in vitro? Because the fertilization process is taking place outside the body of the mother in the laboratory. So that's why. Next, moving further, in vitro fertilization, the embryos are formed by the fusion of gametes. I've already told you about it, and it helps to assess those females who cannot conceive. So sometimes what happens is the female's womb will be in very good condition, the uterus will be in very good condition to bear the child, but the female will not have a good quality egg. So that time what they do is they take the egg from another female and then they take the sperm from the husband and they fuse it in the laboratory. So those women who have a very good uterus to bear a child but they don't have a proper quality egg can go in for this particular method, that is in vitro fertilization. So next is ZIFT and IUT, that is intrauterine transfer. So ZIFT, so what happens in ZIFT we will see. So ZIFT is nothing but the zygote or the early embryo. So in ZIFT, the early embryo or the zygote that develops to 8 blastomere stage, that is transferred into the fallopian tube. Now here what they did, they directly transferred the zygote into, or they transferred the embryo into the uterus, right? But here what they do is they wait for the zygote which is in 8 blastomere stage. That again here the same thing is done. The sperm is collected from the male, the ova or the egg is collected from the female, then both are fertilized. When they fertilize, what will happen? Zygote will develop. That zygote, when it undergoes divisions and it forms a 8 blastomere stage, that 8 blastomere stage that is there, which is an early embryo, that is put into the fallopian tube. Now, it is not put into the uterus, but where it is put into, it is put here into the fallopian tube. Now, here in the fallopian tube itself, it will become 8 cell stage, 16 cell stage further, and that once it matures, it will come and from the fallopian tube, it will travel and it will embed itself in the endometrium of the uterus. So that is what is done here. So don't get confused between IVF and this particular procedure. In IVF, directly the zygote, once the zygote got formed, it will be inserted, or once the embryo develops, it will be inserted into the uterus of the mother. But here, the zygote that will develop when it reaches the 8 blastomere stage, that is inserted into the fallopian tube of the mother. Okay, from there, it will, after it becomes the 16 or 18 cell stage, it will travel and it will come and embed itself in the endometrium lining. So when the zygote or embryo is more than 8 blastomeres, it is directly placed in the uterus. That is called as intrauterine. Now in ZIFT, what I told you, the 8 cell, when the zygote is in the 8 cell stage, it is put in the fallopian tube. Okay, but in intrauterine transfer, what happens? The zygote will not be of 8 cell stage, but it will be of either 16 cell stage or 18. So when it is that condition, it will be directly put into the uterus. So that is called as IUT, in intrauterine transfer. In intrauterine, it is the 16th cell blastomere that is put directly into the uterus. That is put directly into the uterus. So that is called as intrauterine transfer. What is ZIFT? In ZIFT, the zygote which is 8-celled blastomere. So here, 8-celled blastomere, that will be put into the, it will be directly sent to the, injected to the fallopian tube. Then here it will become the 16 cell stage, then it will come here and it will embed. So that is the only difference between zygote intrafallopian transfer and this intrauterine transfer, IUT. So next is gift, that is gamete intrafallopian transfer. So in gift what they do is the ovum that is collected from the donor. Now the donor will be a female, right, because it is the ovum which can be collected from the female. That ovum is transferred into where? Into the fallopian tube of another female who cannot produce one but can provide suitable environment for fertilization and for its further development. So what they do here is they will directly transfer, that is the healthy egg. So healthy egg, they will take it from one female. So this is a female. From one female, they'll take a healthy ova and they will put it into the fallopian tube, inject it into the fallopian tube of another female who wants to get pregnant because she is not capable of producing a healthy ova or a egg. And next, later on, the egg, when it is released, when it travels through the fallopian tube and proper copulation occurs, the fertilization process will happen. So that is about gamete intrafallopian transfer. The same thing can be done here in the case of male also. This is the case of female. In the case of male, what can be done in gamete intrafallopian transfer is, the husband is not capable of producing healthy sperms, then the sperms can be collected from a donor and that sperms can be sent or injected in through the vagina so that the sperms will travel and then it will fuse with the egg that is produced by the female. So both ways it can be done. That is why it is called as gamete intrafallopian transfer. Transfer of sperms into the uterus or into the fallopian tube, or transfer of the ova, healthy ova, into the fallopian tube. So that is why it is called as gamete, wherein male gamete and female gametes are used here. So next method is intracytoplasmic sperm injection. So the word itself says intracytoplasmic sperm injection. So in this what they do is, a single sperm that is collected from a healthy donor is directly injected into the ova using a micro needle. So some of them, they'll have some infertility conditions, they'll have some infertility conditions or genital problems and all that. In such cases, the sperm can be extracted from the husband if it is healthy or from the donor, and directly it will be injected into the ovary or into the ovule. So into the ovule, that is ova or the egg that is present inside the female's ovary. So direct injection will be done to the cytoplasm of the ova or the egg that is there inside the female. So that is nothing but called as intracytoplasmic sperm injection. So in this method, a single sperm that is collected is directly injected into the ovum using a micro needle. And this method is used in case of the male infertility, wherein the sperm count is very, very low. So when the sperm count is very low, or if the sperms are not that active, it is difficult for the sperms to travel towards the egg and fertilize the egg. So at that time what they'll do is, they'll collect the sperm and they'll directly inject the sperm into the cytoplasm of the egg where the fertilization process will take place. Only single sperm which is healthy and active should be inserted into. So that is how it is done in intracytoplasmic sperm injection. Next is artificial insemination. So in this technique, the semen that is collected from the husband. Semen is nothing but that jelly-like mass which contains a lot of sperm. So that semen is collected either from the husband or from any healthy donor, and it is artificially introduced into the vagina or into the uterus of the female. Now the sperm from there will travel towards the released egg which is there in the fallopian tube. There it will fuse with the egg and the fertilization process will take place. So this is called as artificial insemination. This is also a kind of intrauterine insemination itself. So this method is used in infertility cases when the male partner is unable to inseminate the female partner due to very low sperm count. So this also can be done when the sperm counts are low. Either they can go in for intracytoplasmic sperm injection, or they can go in for artificial insemination. So this is about artificial insemination. So next we'll talk about the disadvantages in ART. So we got to know about the advantages. One thing is, it is one of the best procedures for couples who are not capable of bearing a child because of infertility reasons or some other reasons. Now, what are the disadvantages? Disadvantages is it requires highly extreme precision in handling, and therefore very few specialized professionals are there who are capable of doing all these assisted reproductive technologies. So we have to depend on only those high professionals for getting this inseminations done, or getting these reproductive technologies done. And it is affordable only to limited number of people, because the cost of this particular reproductive technologies is very high, and also the reproductive technologies, the success rates are very low. 1 or 2 attempts you will not get a success. Sometimes it might take 3 to 4 to 5 attempts. So it's a lot of money should be spent on this, which everybody cannot afford. And also it will have a toll on the emotional, religious and social factors of the individuals, because every time they'll have to go in for checkups, they'll need to keep visiting the doctors, go in for hormone therapies and all that during this process, and they'll have to take a lot of injections, hormone related injections and all that. After taking all this, if the attempt fails, then it will bring a lot of emotional stress among the couples as well. So all that emotional stress, and also some people will be so religious wherein the family members will not be allowing the couples to do that. All that will lead to a lot of tips or stresses in the family. And also, when all such things, so say for example, some people are not capable of going in for assisted reproductive technologies because of their emotional point of view, or because of their lack of money and all that, they can always have an option. So they can go in for adoption methods. So these are some of the disadvantages in assisted reproductive technology. So this was about the session wherein we completely learnt about different assisted reproductive technologies that are available and how exactly they are done, and in which cases they are preferred. So I hope you understood the session well. So with this session, the chapter comes to an end. So we shall meet again in the coming session talking about or discussing the new chapter. So see you in the next session. Thank you.